The rhythm of your feet pounding on the dirt floor beneath you, the rush of adrenaline after you turn every corner. You're so close. All mazes have an answer, right? You stop and you look further ahead. A dead end. What do you do when you get to a dead end? You just keep going and going? I mean, what else can you do? You can fill in 20 maze puzzles, so you can try and try again, but nature doesn't function like that. Nature isn't yes and no. Dead ends happen. The pressure builds up, and you stop. Let's look at humans. The persistent drum in your chest. Da-dum, da-dum, da-dum. Doesn't stop. It almost never does. But there is a nuance to that statement, and metabolomics is all about nuance. All sickness eventually makes your heart, the sort of finish line, stop. It comes to an end, your life, when the sound of the drum fades off into the distance. You hear the stories of fatal car accidents, cancer, cystic fibrosis, but what about the diseases that start from the finish line? We would think that by now we would have these figured out, right? But no. Scary statistic, 21% of deaths happen because of cardiovascular diseases. And there's so many more scary statistics. We've heard them all. Heart disease is the biggest problem. And today, it isn't just the elderly. It's everyone. Let's get acquainted with a patient. A 60-year-old female diagnosed with stage 1 hypertension. The top number in the hypertension diagram is the systolic blood pressure, aka how much blood pressure is your heart pushing out when it is beating, and the bottom number is diastolic, what is happening when the heart is at rest. So a normal child has systolic blood pressure of 95/100 and a diastolic blood pressure of 80/89. The patient in mind had a systolic blood pressure of 140/159 and a diastolic blood pressure of 90/99. What was recommended? Lifestyle. That's what the patient heard, but did not understand. What about the countless hours spent running, playing, and devouring the food their health nut parents prepared? And although the patient was a chocolate enthusiast, aren't most of us? It was a square diagnosis. You were said to have a balanced life, you were said to not be eating right, not being active enough, but this isn't applied to everyone. They're telling us to live a balanced life, but aren't all diseases just a recommendation of living a balanced life? How do we specify this? Let's focus on one of the most common malfunctions, hypertension. It is one of the most common cardiovascular diseases, and it is characterized by an increase in blood pressure. Blocked arteries happen, which builds pressure on the arteries and therefore creates a difficulty regulating the blood. There is no effective treatment to one of the most common cardiovascular diseases. Let that sink in. Circadian rhythms. What are they? First of all, they aren't just about sleep. Circadian rhythms play a fundamental role in the progression of circadian and cardiovascular misalignments. Almost all cardiovascular diseases have these misalignments, and they're usually characterized by changes in metabolites, an immediate end product of metabolism. These rhythms are essentially a giant clock, just like this one. This is a simplified diagram. We're going to be focusing on the highest blood pressure because we're looking at hypertension. This is approximately around 7 p.m. Circadian rhythms function on a hierarchy. A master clock is a regulator, a project manager of these cellular clocks and metabolites. We can think of master clocks like a battery to a regular room clock, like this red one right here. It is the battery that powers everything else. It regulates everything. As we move down, we can understand that this battery, the SCNs, the specified neurons in your brain that make this master clock, help regulate the cellular clock, the hour hand, and the metabolites, the minute hand. Cellular clocks. They are organisms' natural and specified clocks that are usually found in cells and your organs. They're everywhere, and they're composed of specific metabolites, molecules, and proteins that interact with cells throughout the body or that specific organ. Nearly every tissue and organ has and contains biological and cellular clocks. Now, the process of establishing these clocks and building your way up to the master clock involves metabolites. Metabolites are what we can use to really understand and look at the correlation between SCNs and metabolites and therefore circadian rhythms and hypertension. Let's play a game, a basketball game. In a basketball game, it comes down to two entities: the point guard, the person that regulates the game, and the shooters, the people that finish off the game. Depending on who's in charge, one of these players contributes to something, but eventually it would be the composed entity of shooters because it is a plural, right? Think of the point guard as SCNs and the shooters as metabolites. Whatever the shooters do influences the game. Whatever the point guards, aka the SCN neurons, do, it influences how the shooters would work. And whatever the finishing player does, it creates success. How do we look at these metabolites? The question is when. We can't just be treating the symptoms of hypertension. No, we need to diagnose hypertension by circadian rhythms, having a dynamic and active diagnosis and therefore treatment. My idea is to diagnose it through identifying the correlation between circadian rhythms and the misalignments. I spoke with a mentor of mine, Dr. Duck Youngpon. He's a metabolomics scientist at the University of Frankfurt, and the key and pivotal part of my journey and experience with him is that he suggested LCMS, an analysis tool that separates liquids and solids in samples. Additionally, he has been studying this for 3 years, so it was definitely helpful learning about utilizing LCMS, an analysis tool that helps sample normalize. So how and what did I do? Well, I looked at a procedure of a study and I analyzed it. I looked at the time interval definitions, which were over 8 weeks, and I understood that this is probably normal. I then looked at sampling between SHR, one species of rats, and WKI, another species of rats, which mean super high intensive rats and WKY rats. So the light samples were on for 12 hours and 12 hours off for the rats, and there was a chow and water daily intervention for 8 weeks. But there was something missing. Of course, there was metabolite analysis, but there was no liquid chromatography mass spectrometry. What I meant by that is LCMS. So I redid the procedure and included this as well as a pathway analysis. To test out this procedure in applications with hypertension, I stimulated a metabolomic pathway in pathways. Specifically, a metabolomic pathway of acetonitrile, one of the main chemicals used to differentiate between solid and liquids in a sample, for instance, fatty acids and blood. This is one of the main chemicals, so it is crucial to understand the way it differentiates and the way it can impact the entire study, because how do we ensure a quality data analysis? Additionally, I went on an analysis software and found ideal circadian rhythm intervention time for this study that I'm designing. So if we look at this graph here, we can see that every 16 seconds, as identified by these high scrapers here, we can see that it would be an ideal intervention time. But they repeat twice. You go up and down twice. So it would be 16 times 2, aka 32. So from this graph, we can identify that every 32 seconds, it would be an ideal circadian rhythm intervention time in a study. When it comes to context gaps, we don't only have to rely on the why now, but the how and the when. Metabolomics is an active process. Bioinformatics is an active process. Questions need answers, but those answers, although unattainable right now, can be looked at differently. These topics are interconnected and complement each other. It comes down to the crossroads of bioinformatics and biotechnology. Before I end, how many of you have parents, family members with high blood pressure or any type of cardiovascular disease? Think about that and the way that can be influenced.